Good morning and welcome back to the channel, or welcome to the channel if this is the first time you've visited. You're with StudentSOS. I am once again making a video where I am not in front of the camera, which I'm delighted about, and my colleague who uploads these videos will make sure that I'm not delighted about when he's had a chat with me. Sorry, Chris. So this morning we're going to carry on, having looked at DNA and how we make it into protein. We're going to take a little look at the structure of DNA in a bit more detail. So here we go. On the left-hand side here, we have a double helix. This was identified and first characterised by Watson and Crick. And they, with the help, by the way, of Rosalind Franklin, if you've never heard of her, I understand why not. If you haven't, please Google her. I'm going to tell you her name again. That's Rosalind Franklin, without whom the double helix would never, or certainly not as quickly, have been established. So Watson and Crick were originally credited with this work, and Morris Wilkins shared Rosalind Franklin's X-ray crystallography. If what I've just said sounds like utter nonsense to you, please go and Google it and read about this lady. She was fundamental in the identification of the double helix, and although it was subsequently rectified, she was written out of the structure of the double helix to begin with. Okay. So the most important things for us to think about here are base pairing rules. So in the previous video, I very briefly mentioned the bases involved in DNA and in mRNA. And the base pairing rule that you should know is that adenine, big letter A, pairs always with thymine. Please make sure you are checking your spelling of both of those. Adenine is not adenosine. That's something completely different. So adenine is the base, and thymine is not the same as thiamine, which is vitamin, I think, B12. Please don't comment below if that's wrong. So it's thymine. If you spell these incorrectly in the exam, you will not get the mark. Cytosine, C, pairs with guanine. There's not that many things that you could misspell either of those for. Cytosine, I suspect you might misspell for cysteine, particularly if you're an A-level chemist or know your protein structure super well, but it's cytosine. You'll notice that there's a different number of hydrogen bonds between these two pairs. So you have a double hydrogen bond between A and T and a triple hydrogen bond between C and G. This is always the way these guys bond in the double helix of DNA. The purines, if you've heard this language before, are A and G. I always remember that they're the purines because they are the larger structures. They are the two ringed structures. And C and T are the pyrimidines. And for the exactly opposite reason, I can remember that the long name, the pyrimidines, is what's given to the shorter or smaller single ring structures. By the way, most of my videos are no edit unless it's a catastrophe and then I start all over again. So for your purposes, no edit videos. So one purine pairs with one pyrimidine, and that gives us one base pair. So we're going to take a little look that DNA is found in chromosomes in mammals. I do find this very interesting that even at the end of year 12 often, students are not quite clear what the layout of this is. So DNA is the molecule, it falls under the category of biological molecules. And a chromosome is a way of packaging some DNA. Of course, in humans, we have 23 in normal body cells, pairs of DNA, but different animals have different numbers of chromosomes and different numbers of pairs of chromosomes. So a short section of DNA within a chromosome is called a gene. A gene is made up of nucleotide bases. These are monomers. We're going to have a look at the nucleotide in just a moment, but these are called monomers. We've looked at monomers in biological molecules. We'll be doing a video on that quite shortly. So each gene codes for a protein. Now I'm going to tell you now, it's slightly more interesting than that, but for our purposes in year 12, each single gene codes for a single protein. So here we have a nucleotide monomer. You could be asked to draw this in the exam. You may have done a test with this nucleotide monomer diagram previously. So we have a phosphate, and that comes out of the fifth carbon on a pentose sugar. So in DNA, the pentose sugar is deoxyribose, and in RNA, the sugar is a ribose sugar, but they are both pentose sugars. Useful information if you're asked to look for similarities and differences between DNA and RNA structure, another common Year 12 question. And then we have our nitrogenous, just means nitrogen containing, guys, base. So now we have our A, T, G or C if it's in DNA, and we're assuming this is a DNA nucleotide. The difference then, if it's an RNA nucleotide, is that we'll have a ribose sugar as our pentose sugar, and indeed uracil, U, will replace thymine, T, always in RNA. So as I've mentioned, a nucleotide is a monomer. Its DNA is formed as a polynucleotide of two strands, and they run in opposite directions. We refer to this as anti-parallel. When we take a look at transcription and translation, we will take a look at the 5' to 3' and 3' to 5' lengths of DNA. If you do not recognise that language, go and have a read of that straight after this video. So much like all the other biological molecules, condensation reactions link the sugar to the phosphate group, and they form covalent bonds, and these are called phosphodiester bonds. There's some really useful language to use. There's some money marks for you, Year 12. And these form and break during DNA synthesis and DNA breakdown. So that's the basic structure of DNA, and what we will look at next in the next video is how DNA replication occurs so that we can make new cells. Have a great day. Thanks for watching, and as always, please like, subscribe and comment below if there are any questions on this video, or indeed if there's any content that you'd particularly like us to take a look at. Thanks very much, guys, and I'll see you in the next video.